My name is Yiyang Gong. I'm an assistant professor here at Duke University in the Department of Biomedical Engineering, and I lead the lab that's being funded by the Beckman Young Investigator Award. Our work really tries to follow the same ethos of the Beckman legacy, which is to create a suite of tools targeted to allow neuroscientists to be able to do new science and study the brain in new ways.
I'm working on expanding the genetically encoded tools that we use to study neuroscience.
I'm building a microscope that can achieve large volume brain activity imaging over multiple neural populations over large volume at fast speed and high resolution.
We're using an optical microscopy with the planar illumination so that we could slice the brain fast.
I'm working on image processing, so mainly on segmenting neurons and detecting the neural activities.
Arnold Beckman is first and foremost known as being a tool developer and a tool disseminator. His earliest instruments enabled scientists to do lots of activities that weren't possible before.
Today, many neuroscience use microscopes and genetic tools to image many neurons and get their activities on the timescales of tens of milliseconds. So we need an efficient algorithm to process these videos as quickly as possible so that we can obtain real-time information about the neurons.
So neuroscientists have been clamoring for a fast set of software that can retrieve for them the neural activity quickly and accurately. Our software is actually very simple, but very sophisticated in that it can now process neural activity in an online way. So as soon as the experiment is done, we can actually extract the neural activity.
Because of the Beckman Foundation, I was able to do my research and build this software and give it free to the entire neuroscience community. This software can work well on a $3,000 computer. So compared to other software that requires a computer cluster to get a good performance, which takes tens of thousands of dollars, the hardware cost for the users will be much lower.
On the protein tool side, I think we have developed new protein sensors and actuators that can either record from the brain or stimulate and perturb neural activity. And we can now do this in two color channels simultaneously.
One of the key benefits of the types of tools that I'm developing are that we can use genetically modifying techniques to target them to specific types of neurons. So throughout the brain, there are different types of neurons that are almost always spatially overlapping. And in order to figure out how these spatially overlapped populations are communicating with each other, we need to be able to have tools that can independently and accurately record from each of them at once. So the major benefit of the tools that we develop is that it expands the spectral diversity of these types of sensors. So the benefit of this spectral diversity, i.e. like the different colored sensors, is that we can put one color sensor in one type of cell and another colored sensor in another type. And then we can basically just use filters to separate the signal from each of these populations and see how they're interacting.
So neuroscientists are always trying to image as many neurons as possible to better understand the mechanism behind the brain function. But the thing is, the image volume is always limited by either the imaging region of the microscope or the resolution.
Our optical microscopes are also new. They will now enable a suite of volumetric imaging experiments that allows us to look at large portions of the brain or entire brains, for example, in small organisms like zebrafish, all simultaneously. And we can also do this quickly at the millisecond timescale. And what this will allow us to do when coupled with the protein tools in our protein section is to investigate neural activity at the millisecond timescale.
The challenges we are facing right now are that the brain is a three-dimensional value, and the signals get encoded and transferred in these three-dimensional neural circuits really fast. So we are developing a system so that we can record the activities happen in the brain really fast. and the images we collect should be clear as well, so that we could apply post-processing algorithm to detect the neurons and identify the activities easier.
So overall, the hope is that by producing these tools, disseminating these tools, and enabling the neuroscience community to perform new experiments, we're going to be well positioned to carve out a space within the scientific world that can always push forward the understanding of the brain.
I greatly appreciate for the Beckman Foundation to provide us the opportunity and access to a lot of state-of-the-art equipments.
Whenever we talk about this work at conferences like the SFN conference last year, a ton of other people in the neuroscience community are really excited to hear about how these voltage indicators are ever advancing. And I think that as we continue to work on it and continue to expand their capabilities, we'll be able to discover some really cool and exciting things.
In my own history, I've been working on optical microscopy and protein tool development for about a decade now. And with the entire suite of tools that we've developed under the Beckman funding, I think we're hitting an inflection point where we can really produce large-scale experiments that's going to greatly enhance our understanding of the brain going forward.